All right, so while people are joining, so welcome everyone to this week's Theoretical and Computational Biophysics seminar. It's my great pleasure to host remotely Professor Ivet Bahar from University of Pittsburgh. Before I start my introduction, I invite everyone to turn off their mics and turn on their cameras. That's always encouraged to give a more interactive feel to the seminar. So let me go ahead with a formal introduction. By way of introduction, I guess many of us know Ivet very well, but by way of introduction, so Ivet received her initial training as a chemical engineer in Turkey, Bogazici University and Istanbul Technical University in Istanbul. And then actually she joined the faculty there, and she stayed there for a few years where she went through the ranks and she became a full professor before she moved to this side of the Atlantic Ocean actually to join the faculty at University of Pittsburgh. Initially at the Department of Molecular Genetics and Biochemistry for a few years, but then she became the founding chair of two departments actually during her tenure at Pittsburgh, departments of computational biology and computational and systems biology later. So she founded these two departments and she really boosted the computational biology at Pittsburgh to a significant degree. So currently, she was also founding director of the Center for Computational Biology and Bioinformatics, and also initiating a PhD program in this area at Pittsburgh. So currently, she is a distinguished professor of computational and systems biology, and also associate director of the Drug Discovery Institute since 2010, as well as the director and PI of one of the BTRCs of Biomedical Technology Research Centers of NIH that we know very well, BIOS. We have had actually a lot of interactions with Ivet and her center at Pittsburgh. So this is another P41 center, serves the biomedical community there. So she has a very long list of distinguishing awards and recognitions. I don't want to actually steal from her talk's time and to go through them. I only notice, and again, many of us know that just last year she was elected to the National Academy of Sciences, recognizing her major contributions to the field of computation and systems biology, I should say. I also noticed that you have been an invited speaker at the White House, again, contributing actually to the strategic computing initiative of that body. So she has served a lot of communities and societies, Biophysical Society, EMBO, and also elected member of the National Academy of Science in Turkey and other countries. And in terms of her research interest, she has really broad interest in computational biology. Obviously, we are going to hear more about her interest in molecular sciences more today. But beyond the protein dynamics and modeling that she's famous for, the methods and the tools that she developed for the community, she's also interested in druggability of targets in the cell and also cellular modeling. So these are among the missions of the center that she's directing. And being affiliated with the P41 Center, she's very aware and she has been very cognizant of the importance of service streaming in the community. We have been actually giving joint workshops since a few years ago, and I look forward to the next one, maybe in person again. Thank you very much, Ivet, for accepting our invitation and joining the CCG seminar, and we look forward to the seminar. Please.
Thank you for the kind invitation and introduction. It's a pleasure to be here, and I see many familiar faces, so it's a great pleasure to catch up. So today I'm going to talk about a few recent developments on elastic network models, and I slightly changed the scope to fit it in 40-45 minutes. So I'll talk about the essential dynamics and how we generate conformational landscapes for proteins or biomolecular assemblies. So here is the plan of my talk. Do you hear me well to begin with? Okay,
yes.
great. So I'm going to start with a very brief overview of elastic network models. I assume that by now most of you already know, but just for completeness. And then I will move to hybrid methods, how we combine elastic network models and molecular dynamic simulations to try to benefit from the best part of both methods. And then a new ESA is essential site scanning analysis, how we predict essential sites using ESA. And finally, how we use multiple tools in a protocol for discovering drugs that target allosteric sites or allotargeting. So elastic network models, many of you already know, we just represent the structures in terms of nodes and springs. And then using that representation, we try to see, we move the molecules in the space of collective coordinates, which means everything moving simultaneously in a cooperative way along the modes that are intrinsically accessible, and then try to make inferences on functional mechanisms. In a nutshell, this is the summary. So a little bit more basic description. We usually start with a known structure. So we're not solving structure. We are just analyzing the equilibrium dynamics of existing structures and trying to find what are alternative states, substates available for achieving function. So we map a given ribbon diagram, usually a ribbon diagram, into a network model composed of nodes and beads, beads and springs. And here is a brief summary where a movie from a student who shows you the procedure. You start with the ribbon diagram. We focus on the backbone, and alpha carbons are each represented by a bead in the simplest elastic network model. And then those within a certain cutoff distance, a first coordination shell, are connected. And then we obtain a network whose normal modes are being analyzed using basic solid state physics concepts. So that is what the most commonly used anisotropic network model is about, essentially starting from a simple representation, trying to dissect the dynamics. And here we can see a few movies for an example protein, HIV reverse transcriptase. And we have a hierarchy of modes or motions that are accessible. First mode, second, third, fourth, etc. And each mode we are showing here as a color-coded diagram where the red portions move a lot and the blue portions, they are served like anchors or hinges mediating the collective motions. And everything in this approach is actually defined by the contact topology. If we know what we call the connectivity matrix, which residue pairs make contacts, we can build a Hessian, assuming harmonic potential between all connected pairs. And this simple Hessian can be represented in an analytical form. So we just solve analytically for the spectrum of motions, the mode ensemble of modes, and then we can, generate very easily this type of movies for each protein examined. In this case, we're talking about the Gruyère chaperonin, viewed from top, we have two cylindrical portions that undergo counter rotations. Here is a membrane protein, AMPA receptor, tetrameric, and here is gamma secretase. And as you can see, depending on the architecture, the proteins will be favoring different types of cooperative motions. And the idea is the fundamental assumption, if you want to take a message, is that these structures have evolved to favor motions that are functional. So when we solve analytically the favorable movements, modes of motions that the structure uniquely defines, they ought to be meaningful in terms of biological function, because that is how those structures have evolved. So that is why we want to understand what those motions are. And we can do that using the PDB structures or cryo-EM structures, just using electron density maps also. We know what are the collective movements for those of you who are more interested in cryo-electron microscopy. The good news is that we don't need a high resolution representation, even a full atomic representation. We can, carry out this type of analysis easily, even with a coarse-grained description of the structure. There is an interface we put together, ProD, for analyzing protein dynamics. So usually users may give a sequence, if you wish, and from the PDB, ProD is going to extract all the structures that match this sequence with a certain sequence identity threshold, which you may also define as a user. And then we get an ensemble of structures. The ensemble of experimental structures can be analyzed by principal component analysis, or a representative structure can be also analyzed to find the motions. And we can compare the principal components from experiments or the normal modes from theory. Oftentimes we see some correlations. Sometimes the order of the modes changes. And then we can see what is the space of experimental structures, those predicted by the network model. Here, ANM is an isotropic network model. And then we can compare to MD simulations, etc. And this interface has been very broadly used since its inception in 2011. We have recently published the latest version for those of you who are interested. Ivet Bahar, a former PhD student, was the force behind it. He is now working, he became a senior investigator or programmer, I don't know, software engineer at Google in San Francisco. We also put together another interface called Dynamics, actually for biologists, because ProD requires Python knowledge. So some minimal programming, if you wish. That is totally intuitive for users. You can just enter your PDB code, and you can include the environment like the membrane, or if it's a part of an assembly, you can put the remaining molecules, and then you obtain similar results, movies. After all, we are turning static images, structures into movies. That's what we're doing. And then we can analyze what are the critical sites, cross correlations, etc. So here, pros and cons of ENMs, elastic network models. I already talked about the pros: a unique analytical solution, very efficient, can be applied to multivariate systems. Global motions, it means, opposite of local, whatever involves the entire architecture. We can easily characterize them and then relate them to function, extremely efficient. And because it is, instantly we obtain those analytical solutions for a given system, we can use that in a proteome scale machine learning algorithms as features. As to the cons, disadvantages, we have no atomic detail. We cannot accurately model any local events. We just find accurately domain movements, but not local rearrangements usually. So there is no specific residue specificity. For example, if the structure is retained, we cannot distinguish between different types of amino acids. No energetics, everything is assumed to be harmonically bonded. And then we have, the energy surface is approximated by a harmonic potential, no ruggedness. It's a good and bad thing at the same time. And then we can't apply to non-equilibrium events like unfolding, binding, substrate, ion channeling, etc. So we would like to use the best of both worlds, let's say, and design methods that would be taking advantage of both ENMs and MD simulations. Now, in the case of MD simulations, it's funny because you can change all the pros to cons and all the cons to pros and just put no in front of everything. And this is literally valid. So of course, you have atomic models, you can apply to local events, non-equilibrium events, etc. But they are not efficient, and they are, the global motions, there are sampling inaccuracies, etc. So we have come up now with hybrid methods to address the limitations of both methods, hoping that we will be able to predict better. So that one of the early studies, we thought we made about more than a decade ago was, okay, suppose we consider the energy, the conformational landscape. There is an energy minimum here at the state S1, and S2, S3 are like 3 different substates, if you wish, of your protein. And, if you do some kind of experimental, the analysis of existing experimental structures, you would see these are the principal components, the major displacement and another one. Now, if we do our own elastic network analysis starting from S1, we obtain also 2 major modes of motions, let's say. One of them is very likely, and we got, you might think we got lucky, but usually one of the modes will be moving, letting the original structure move along another substate. And the second mode could come close as well. So this is not exactly getting lucky, actually. As I mentioned, the structure is such that it should favor alternative substates in order to be functional. So we have observed that in hundreds of applications. And then we said, but we are not able to actually, see if there is an energy barrier between 2 substates. What the theory elastic network models are doing is will be just fluctuating in a given minimum and would not be able to surmount an energy barrier, or it really approximates everything by a very coarse-grained shallow minimum. So to move from one state to another, to explore the space in between, we came up with an adaptive A&M in 2009, and it is still very broadly used. The idea is you start from one structure and you predict a number of, a certain number of modes to define your first step, a collective step, if you wish, from one end, and you do the same thing from the other end. Now you have 2 new endpoints, and then you have new steps, etc. This way you are able to moving in the space of collective modes, which means you take always the slowest modes, the smoothest passages, the passages that require the least energy, the least energy ascent. You may find a path to move from one to another. And if you compare to interpolation, this path has much, much lower energy. But you can define some, parameters, whether you would like to move in very small steps, and you would like to depart, how much you want to depart from the targeted direction, etc. But this adaptive A&M is useful because when you get to a given new point, you reevaluate all the modes. And this way, if the structure has deformed to a certain extent, some pairs of nodes are not anymore close enough to be connected. So you can, dissociate the structure or really sample alternative conformations. Otherwise, you are stuck in the neighborhood of the same minimum. So that doing that, for example, we were able to simulate for AMPA receptor as an example, the passage from its N-shaped form, it's called N-shaped, to O-shaped. So remember, this is a tetramer. All the monomers are in different colors, and this is the top view. Now, during the motion, this is the N shape, you see, and then it becomes like the O-shaped. Can you follow that? So it is from the top, you can see how the four monomers are moving apart and then collapsing again into, and there is one mode that actually enables that. One of the modes is, as you can see, the top view, it goes from the N shape to the more compact shape. But it is when you do through A&M a succession of movements, you reach with, within atomic resolution, you reach the target as a succession of collective motions. Another, so this was 2009, but even before that, with Emad and Dr. Klaus Schulten, we have built one of the first A&M guided hybrid models, where we have defined the target conformation using, again, elastic network model modes, and then we try to push in that direction the molecule using targeted MD. Another approach is in 2013, we did with late Jeff Madura another approach where we have, it's called co-MD. This is very similar in conceptually. You are just, what you're doing is at every single step, you're moving in the space of collective modes. Instead of moving one atom or one at a time, you're moving all of the residues simultaneously. And then because now you have deformed the structure, you perform a short energy minimization to calibrate, correct for any unrealistic deformation, and then you do it again in an iterative way. So that is, this way we can easily generate such conformational landscapes. One application was to annihilate kinase, a classical example, and you can see the path from open to the closed form. What are the successive movements? Okay, so there are many, many others, and I have to say that this type of hybrid methodologies, they have not really come of age. We have in a recent article published in Current Opinion in Structural Biology, we gave a brief overview of what are existing hybrid models that combine elastic network models with, let's say, Monte Carlo simulations, Brownian dynamics, MD simulations, etc. So at the center here, you see the full force-based normal mode analysis, at full force field-based normal mode analysis. So you see all the methods you are familiar with in the center. And then, although this diagram looks a little bit complicated, suppose, okay, let's consider this EBDMIS. It used Brownian dynamics, and also it used elastic network models. You see, it comes from here. And this is what Modesto Orozco and Eric Lindahl did for predicting the transitions in a study in 2016. Here, this is from Martin Zacharias. He has been using E&M replica exchange MD. So as the name indicates, he combines replica exchange simulations and E&M-based NMA to predict this type of large conformational changes. So there are many, many different labs, including our own lab and others. This is the application to docking simulations, for example, by Alexander Bonver, where they have been generating an ensemble of conformations to facilitate to generate docked conformations of, in this case, protein DNA pairs, but in general, it's very easy to generate an ensemble of conformations using these elastic network models, which you can use it for many different purposes. MDENM is from David Perahia. I'm going to come to that one. So overall, the reason, what I would like to emphasize is that ENM-based normal mode analysis has been a tool very broadly used in many hybrid methods in combination with full atomic simulations. After all, it proved useful for modeling multi-scale events, a thorough sampling of conformational space, understanding mechanisms, and also, as I'm going to mention, describe identifying small molecule modulators of functional interactions. Okay, and so this is the review that I just mentioned for those of you who would like to see the literature. And I'm going to describe very briefly the latest hybrid method that we have introduced, actually based on a paper published a couple of years ago, 5 years ago already. Now we have the interface and the more established version ready to be used by the community, which has just appeared. It's called Clust ENMD. A cluster comes from clusters ENMD. You can imagine now, you understand what we are talking about. Now, this is a hybrid technique which samples the conformational space and easily applicable to multimers, assemblies, fully automated, available in Prodi. As the MD software, we're using OpenMM, and we need to use an MD. So we need to work together to do that. And it is very efficient on GPU architecture. So what it is doing essentially is you start from a structure, you generate an ensemble of ANM modes. So it is like generating several generations. So you take a few modes, slow modes, so you generate, let's say, 8 alternative conformations based on 4 modes, moving symmetrically in either direction, because these are fluctuations. And then you start those conformations, you try to cluster them to get some representative conformers to generate a new generation, second generation. The clustering is needed because otherwise you have exponentially growing conformations. And then for those clusters, representative conformers from the clusters, you do an MD simulation or relaxation so as to re-evaluate the ANM modes, and so on and so forth for several generations. So you gradually move away from your starting conformation, and you can visualize, you can use all the Prodi infrastructure for generating movies, etc. So again, the application to analyte kinase allowed us to sample these conformations and those. But here a caveat, we started from this and we sampled these conformations, the dots, the red, black dots. And when we start from here, we sampled this, but we were also very happy to see that there were a few cases sampling this passage, space in between the two states. So we were really happy because we could reach that region. And then originally, the easiest thing to do for us is if we have 2 endpoints, we start from those 2 endpoints and we want to see how the space is being sampled in between. So this is, for example, for HIV protease, the blue dots here represent the experimental closed structures. And here we have open structures, fewer of them. And then if we start from the closed, we generate the magenta, and from the open, we generate the cyan, or you can represent it as a surface here. But to me, what is interesting is when you do this type of computations, and I'm showing you the successive generations, gradually you start sampling the right location between the 2 endpoints. You see, these are some intermediate conformations, and they are, as the surfaces grow and there is an overlap, we can see that this region of intermediates is being covered. So we have been using that not only as a, starting from 2 endpoints and seeing how we can sample the intermediate, but also starting from a single point. And we recently, did a really very extensive study applied to many proteins of 5 different hybrid methods. So one of them is co-MD that I just described, you remember, and the other one is class C NMD, the most recent one. Class C NMD is based on class C NM. There is a small difference. This one doesn't have, class C NM doesn't have energy and doesn't have MD simulations, just a short minimization. That one has longer MD simulations. And then we have the method of Perahia, Acosta, and others, MDENM, where they use full atomic normal mode analysis in combination with MD simulations. And then we wanted to see what are the predictions. And I think this is again HIV reverse transcriptase. What is interesting is HIV reverse transcript, I'm sorry, this is HIV protease, not reverse transcriptase. HIV protease is one of the most widely studied proteins. So it has been structurally resolved in hundreds of different forms. Like there is more than 500 different structures in the PDB. And then when you take those structures and you look at their conformational variability to find the root mean square fluctuations, you can see that here is what you observe, for each residue, how much they move, when you superimpose structurally align those structures. When you do MD, it moves more, and then in the hybrid methods, we even see larger movements. But what is interesting is if you look at the correlations between those plots, with taking the experimental curve as the basis, you can see that for all of them, all of the hybrid methods, the correlation coefficient is quite high, and it is even slightly better than MD simulations. And in many examples, we've seen a similar behavior. So we can definitely, using those hybrid methods that are very fast compared to full atomic simulations, we can have a good understanding of the fluctuation behavior. And also, here is the projection of the experimental structures shown in cyan to the subspace spanned by 2 principal components, PC1 and PC2, which are defined by the experimental structures actually. And then the cloud here shows the space sampled by MD simulations, by class C and MD, etc. And we can see that usually those hybrid methods will be sampling a much broader space, which would be covering also, let's say, both open and closed forms or alternative states. So we were overall, there is room for improvement for sure, but overall, it looks like we've made a lot of progress. And what is also interesting is, from experiments, again, using principal component analysis, you have the experimental principal component 1, 2, 3, 4, etc. For HIV protease. And now if you use one of the hybrid methods and they more or less give the same result, you can also, for a single structure, you can predict what is going to be the first mode and second mode of motion and third, etc. And then you can see that there is a correlation in between of 0.76 or so. So the first, these experimental ones are the product of hundreds of structures that have been resolved. These are obtained just within seconds for a given single structure. And we can see that, the space covered is very comparable. There is, of course, it's not perfect, but it gives you a good agreement overall. Now, what is ESSA? And we are coming to more interesting parts. ESSA is these new analysis methods also recently published. So we want to understand what are the essential residues. And you may know essential dynamics of Amadei, Berenson, essential dynamics was the terminology they used for the global modes, right? Or the lowest frequency modes. Now we are also essential sites in our case are residues that control the global motions. So that, we were inspired by the same terminology. So what are the essential sites? Of course, ligand binding sites are essential. Catalytic sites in enzymes are essential, or you may have mechanically important sites like the hinge sites. So when we do the analysis is the following: you take a given structure and you perturb each residue. How do we perturb it? Well, we place there the side chain atoms, which is a crowding in that region. And in the presence of this type of crowding in the network model, where now there is an aggregation of nodes in this region, what is the effect on the global dynamics, the frequency of the global modes? That's what we examined. And for each residue, you repeat the same thing. And for each residue, you have a score called z-score. And the idea is that the peaks should be really important sites that cooperatively affect the overall structure. And they may usually contain allosteric residues, but also orthosteric sites, the catalytic active sites. They may be also affecting the essential dynamics if you perturb them. So you have many, many peaks here. And the challenge is to discriminate between them, which ones are really the most important ones. And we have been combining, we can, plot the results here could be mapped into color-coded structures. The peaks are in red, and we have verified for in many, we applied to an ensemble of proteins, and we've seen that the red regions, high z-scores, usually bind small molecules. These are the critical sites because small molecules, they bind regions that would really affect the protein function. If they bind a region, let's say blue, a cold region if you want, they are not going to have any effect. So the logic is that the essential sites would be binding, would be good sites for perhaps drug binding. But among them, not all of them are likely to bind drugs. And we would like to have also some kind of a pocket. So we have used this SI analysis in conjunction with a pocket identifier software. And then we identify really sites that are most likely to be druggable and affect the global motions. Now, when you apply the method to to a series of proteins, you would see that the z-scored histograms for the non-allosteric, non-orthosteric or hinge sites are usually higher than the rest. But still, it's hard to distinguish, right? So that's why we need this type of combination of more than one tool. Otherwise, we are still in the dark, even though we have some kind of a signal. So, for example, this was a nice case where applying by GPCRs, we were able to identify those red regions with high z-scores, essential sites, which indeed bind small molecules, or even the G protein binding site itself turn out. Here you see the GPCR slightly tilted to see the cytoplasmic region where the G protein binds. So that turns out also to be an essential site whose perturbation would elicit a cooperative response. And then you can, if you want, you can try to find what is the communication path, etc. So there are many applications. This is another one. It looks, this enzyme PR65 formed by many repeat units, they all look identical, right? But when you do SI analysis, you locate this region to be the hotspot, if you wish. And this is exactly where the catalytic domain of the heterotrimeric PP2A protein phosphatase 2A binds. So we have seen many such applications. What is also beautiful is that suppose you take beta-lactamase here. Originally, the drug binding site here is not accessible, but now you generate alternative conformations using class C and MD, and one of the conformations really exposes this region. So you can apply, everything is very efficient computationally, remember. You can generate alternative conformations and perform the SI calculations for all of them. This way you can identify a cryptic pocket. This is known actually, but if you start from APO, you can still identify it as one of the class C and MD conformers. So that is another utility. Again, there are advantages and disadvantages. Let's talk about disadvantages because I talked about that. So we don't discriminate between different types of essential sites. And then we use another software, FPocket, to identify, to refine our searches to discriminate allosteric sites. And then everything is based on mode frequencies, but also the mode shapes themselves change. And then we didn't do that. So there is a lot of room for improvement. And I hope many of you will be interested in that for future development of the methodology. One of the pros that I want to emphasize is that the combination with druggability simulations is really very powerful. For example, when you take AMPA receptor, where we did some druggability simulations, we identified many regions that are druggable in our original druggability simulations published a couple of years ago. Now, when we combine with ESA, we can narrow down and identify regions that are really known to bind ligands. So that is, so you use, if you wish, the intersection of druggability simulations, the druggable sites, and among them, which ones are essential, which means which ones are going to affect the global dynamics. And these are the sites that you want to target for allo targeting. Druggability simulations, very briefly, you do your simulations, MD, these are classical MD simulations. And in the solution, you put probe molecules here, examples of them, which are representative of drug-like fragments. And you just try to see where those molecules land on the surface. So you have multiple snapshots from your trajectory, and you just do a statistical analysis of the sites that have a high affinity for binding different kinds of probe molecules, as you can see here. And then you can carry out computations, in this case, cytochrome C, among the different probes. You can see an imidazole always bound to that position. So that's why we ended up by imidazole substituted drugs for modulating the functionality of cytochrome C. We have a few papers on that. So, you can come up with design ideas, and also depending on the type of probes that, stick together in a given site, you can build pharmacophore models. So here's the entire protocol. You start with the structure, you generate alternative conformations, identify pockets, select the essential spots, and then suppose you identified a critical region, essential and druggable. And then you build a pharmacophore model based on the properties of the interactions between your probes and residues, protein residues. And then you can, using this pharmacophore model here, a shape with certain chemical features, you can screen libraries of small molecules and then use other software. Pharmit is not ours, it's from a faculty member in our department, neither Glide, that's also from another group. But you can use existing tools also to screen virtual libraries and do cross-validation. So that has been a very useful tool. A recent application is an application to a GPCR, where there is, this is a class B GPCR, PTHR, parathyroid hormone receptor, which binds a parathyroid hormone somewhere here. It has an extracellular region, and first we analyze, in general, the structure, the allosteric dynamics, etc. But then And of course, there is this TM5, TM6 loop that undergoes a large reorientation. This we all know. But what to us was more interesting was the essential site analysis. You remember the z-scores, which helped us identify a few residues whose perturbation would be critical to affecting the overall allosteric dynamics. Now, of course, these were peaks, right? And very distinctive peaks, but also they coincided with a pocket region, with a pocket region identified by F pocket. So then we said, okay, this is a druggable site, and these are the essential residues, and we would like to target them. So here is also a very nice way of seeing what happens. Now, without the PTH, the hormone that binds, and we do SI analysis, we identify this region to be critical. This region is actually where the PTH binds. So no wonder PTH binds a critical site, right? So this is where the PTH binds. Now we take the PTH bound form and we do the same analysis. And you can see how the structure all of a sudden gets lit, if you wish. Now there's a critical region here, another one there, and here also becoming essential. And this is the G protein binding site, by the way. So we know that binding of this hormone completely changed the cooperativity, and there are essential sites now very distant, even distant from the hormone binding site. Now, after we have identified this region, and then we did also druggability simulations now, identified the probes that bind and build a pharmacophore model, and then identify hits, etc. So based on this type on precise druggability, focusing on the essential site, we were able to discover a few inhibitors that allosterically mediate the function of PTHR. So that is, we think this type of combination of essential sites and druggability simulations with pharmacophore modeling is a very useful tool for future studies. The good news is that everything I explained today, they are available in our PRODI website. There is the class DNMD or co-MD that I mentioned for sampling the space, essential site scanning analysis, DrugWiz for druggability simulations, just preparing the system, and then we use NAMD. And then there is also another component for analyzing the trajectories. And of course, PharmMaker, which is the pharmacophore modeling. Thank you very much for your attention. I hope I didn't take longer than required. And I would like to thank here former lab members and current lab, well, these are all former lab members who have really played critical roles in the development of PRODI or elastic network model-based models. Lee was my first PhD student in Pittsburgh. Zhang was another one who, again, second, he did the adaptive A&M. Ahmed is the originator of the PRODI together with Anindita and Lidio. With Ying, we started to incorporate evolutionary concepts, a new module on sequence analysis combined with structure. Mert is the co-MD guy, and Honchan, James, and John are the latest members who have now left or graduated, and they have contributed significantly to the latest version, especially a new module called signature dynamics, where we analyze families of proteins and analyze how the structure and dynamics evolve. Next time I will talk on signature dynamics, perhaps. Many collaborators over the years, and then here's how my group looks like these days. And thank you very much for your attention. Okay.
Thank you very much, Ivet, for this overview of the state of the hybrid and ENM and other methods. It was very interesting. So before I go into the question, I wanted to point out that we wanted to make sure that you are aware about NAMD3 that we just ported to GPU that has comparable speed actually to Amber and OpenMM. Just wanted to mention it doesn't have all the features and bells and whistles, but for vanilla MD is almost as fast as those two programs on GPU. So just wanted to let you know.
Yeah, we have hard time keeping up with everything you do, Ahmad.
Well, I mean, we are not doing as much as you do, so that's very nice.
Okay.
Thank you. So let me actually start the question myself. So you mentioned the cryo-EM. That was very interesting kind of movie you were showing. So you need to model something, or are you working directly with the density information into those things to
That's
do the,
correct.
I see.
We use directly the electron density maps. And it is interesting because there is a software that originally Klaus introduced, several years ago. And then Jianpeng Ma also used something very similar.
I see.
So one challenge is that we have the shape, but we don't know the connectivity, right?
Right.
So, and then there are, additional tricks to try to identify what could be the connectivity. For example, we need to separate the different subunits of a complex.
I see, absolutely.
So, but as long as the dynamics is pretty much defined by the shape, no matter what the connectivity is, but you have this overall shape, we can make lots of predictions too.
Yes, very nice, very nice.
Yeah, I couldn't talk on that today.
Next time here. So if you have any questions, please raise your hand physically if your cameras are on or on Zoom. Hyun, you can unmute yourself and ask your question yourself.
Hello, Dr. Bahar. Wonderful presentation seminar.
Thank you.
Yeah. So actually, I just want to confirm one thing. So for the adaptive A&M, I think it was,
adaptive A&M. Yeah, that's true.
So just to understand the technique, probably I will read the paper too, but is it like you first calculate the Hessian matrix of the harmonic potential of your structure, and then from there you choose the top k, like whatever 30 like eigenvectors from there, and you add all those vectors up and project back onto the protein space, and then that's what you are showing, right?
That's right. I don't know if I should go back to my presentation, but you have to define each step. And each step is actually a combination of slow modes itself. So there are two parameters that define how many modes you take. One of them is you would like the sum of the modes to be, providing a sufficiently high correlation with the targeted direction. So you take it like 2, 3, or 10 modes to get that sufficient correlation. But you would like also to have very small steps in principle. So there is always a trade-off between speed and accuracy, but the smaller steps you take, the better. And then you keep updating each time. So although we have originally defined for a given endpoint, it is also possible to carry out without knowing an endpoint. That's why we moved to co-MD, because in this case, you start from one conformation and you randomly select any mode, and then you move in the space of modes just randomly. If you have also a target, you can implement their Metropolis algorithm to force it to move toward the target, but you don't have to.
Oh, so that co-MD, cluster-MD, I am a little confused.
I'm sorry, there are many models. Methods.
So, but it seems like it's pretty efficient in terms of like exploring the free energy surface, may I say?
It is indeed, yeah.
Yeah, but one of them was combined with MD though, right? Was it cluster?
Co-MD is combined with MD. Adaptive A&M doesn't have MD. It just has a short energy minimization.
Oh, and when you mentioned the step size, you're mentioning about the RMSD of the protein change in the eigenvector direction, right?
Yes, the eigenvector itself is a vector, right? In the multidimensional space. And then we define also a deformation vector between two endpoints. And we would like to see the correlation cosine between those eigenvectors and the direction you want to move in. I can, feel free to contact me if you have specific questions, and I can give more details.
Thank you so much.
Jale.
Hi, thank you so much for the great talk. It was very exciting.
Hi, Jale.
Hi.
I remember you from last time.
Well, I'm flattered. You have a very good memory. So actually, with connection to what you were just mentioning to Hyun about the A&M guided transition pathway, does the user supposed to accept that step size? Does the user has to do anything else, or you just define the starting point and an end point and the step size?
Yeah. So the good thing about class DNMD, the one I described today, Jale, is that it is fully automated. You don't need to do anything. You just go to ProD, go to class DNMD, and hopefully that's what I think. Yeah. If you start with a given structure, it's going to generate alternative conformations and put together a conformational space for you. It is supposed to be fully automated by now.
Okay. And I have just one more technical question. So I understand that the whole idea of the ENM is more based on the polymer physics. So the range of the interactions that you consider to be about 7 to 10 angstroms, it is more physics-based. But has any study been done on, say, a shorter range for the interactions, for example, 4 or 5 angstrom? I'm asking this because I was just doing over the weekend ENM because I had to compare. The reviewer asked us, can you do Ivet Bahar's method and compare it with your method, which was 4.5 angstrom? And I noticed that, so your actually ENM gave me better results, but I was thinking that can I now reduce the interaction range to say 5 angstroms, which we usually use
So,
or
were you using ANM, trying to do ANM?
I use GNM.
GNM, okay, yeah. GNM is good. With GNM, you can go to 5 angstrom, indeed. But not with ANM. What happens is that, you want your structure to be fully connected.
Sure.
So if the distance, between two residues is too short, there may be some disconnected portions. So that is the bottleneck actually. And the way you can probe whether the distance is too short or not, you look at the number of zero eigenvalues. So when you do GNM, if you have more than 1 zero eigenvalue, then you got a disconnected system. And in the ANM, you are supposed to have 6 zero eigenvalues for the rotational and translational degrees of freedom. If you have more than that, then the cutoff distance that you took is too short.
Okay, thank you so much.
Zoe.
Hello, thank you so much for this wonderful talk. I mean, you've done so much work in the area, and it was, it's amazing to see that it's all now automated and people can use it, students. So thank you so much for the talk. So I had a more general question. Unfortunately, I joined after half an hour, so I'm hoping to see the recording. So maybe you've mentioned this, but through your experience in studying allostery, what would you say are the most important features for studying allostery? So coevolution of residues, motional correlations, I think I've heard. Yeah. What else do you think is a hallmark of allostery, let's say?
You are asking the most difficult question, Zoe.
Yes, my question actually, I was going to ask that last.
This is the most difficult. Everyone, you have, when you speak to different people, many people have their own definitions. Experimentalists will say allostery is all about kinetics. If you have a first binding event, and then the second is going to bind faster for a multimeric system. For example, ATP binding to the first subunit of a chaperonin will be triggering faster binding to the other subunits. And this is, they just focus on the kinetics, and allostery means, or in classical example, hemoglobin, you know, the transition from the tense and relaxed form. There is a cooperativity between the 4 subunits. Now, this is what classical textbooks or experimentalists may tell you. Now, we approach from a more structural or mechanistic point of view. You know, when you map everything to kinetics, a few force constants or rate constants, you don't have information on the mechanism. To us, to me at least, allostery means cooperativity. So an allosteric event is something when you make a change somewhere, like ATP binding or a small molecule binding, and then you have a response at a distal region. So because there is a cooperative response of the overall molecule. And some points on the protein are gonna elicit this type of cooperative response, others don't. So to me, the goal when I try, you know, to identify allosteric sites is to identify the regions that will trigger cooperative responses. And what are cooperative responses? These are in the spectrum of all modes of motions. Some are very cooperative, others are very localized. So I would like to find the sites that are gonna cooperatively change the global dynamics of the molecule per turn. So this is my definition. But we had a workshop, CCAM workshop, a couple of years ago, and a very small group of scientists, and then so many different ideas on even the definition of allostery. And then we came up with I proposed to them, why don't we put together everything we discussed and to say what we understand from allostery. And we put together a review kind of, and the title is allostery in many disguises formed. You know, you can, it is really hidden kind of what an allosteric site or allosteric effect is. And if you are interested, I would recommend you to take a look at that.
Thank you very much.
Many different definitions, but to me, allostery and cooperativity, they go hand in hand.
Thank you.
Emma, did I answer your question too?
Yes.
Well, yeah, sure, absolutely, more than the answer.
Yeah.
So I'm gonna actually ask you one last question myself. So in terms of protein design, have you or other groups have successfully, let's say, went into a protein, have gone into a protein to change this kind of the connectivity that regulates or controls?
No, that is a very, very good point. We haven't done that.
Okay.
We haven't done that. So the really very natural future direction is protein design, indeed.
Yeah, absolutely.
That is something we definitely would like to pursue. We have many studies trying to understand the effect of mutations. There is a new interface. We have this Rhapsody, maybe you know. But that is and this was a machine learning approach, which was possible because we can do high throughput computations using elastic network models. But protein design is something we really seriously would like to pursue.
Sure,
and I can't say we know it.
Yeah. So we stay tuned then.
Yeah.
Any other questions from the audience? If not, please join me to thank Dr. Bahar one more time. Wonderful talk. Thank you very much.
Thank you. Next time I'll be there in person. And Emma.
Okay, look forward to it. So we're gonna stay on the same call. So when people are dismissed, please you can leave. So we're gonna say you kindly agree to meet a couple of group members. Charlotte is gonna be here and Archit, right? Archit.
Yes.
Okay, so Chun, you can leave. Yeah, okay. So while people are, okay.
Okay, we are done right now.
Yeah, so we are done with the seminar.
Except
so now this is the meeting. portion
Yes,
of the.
yes, yes.
Okay, so you can give it, Barry, to Archit or Jale.
Oh yeah, Jale, you're already the host, and you can. I think you can hit the stop recording button now yourself. Don't worry, we'll cut it right for the for public consumption.